phospholipids have a polar head and a nonpolar tail, two phospholipids attached with weak forces to form a bilayer. The polar heads are hydrophilic while the tails are hydrophobic. Polar heads face the outside and inside of the cell to protect the nonpolar and hydrophobic tails from the aqueous solutions on the inside and outside of the cell. Types of proteins incorporated within the plasma membrane. The first type are the integral proteins. Integral proteins are located within the lipid bilayer. These proteins have a hydrophobic region that are made up of nonpolar alpha helix amino acids and a polar hydrophilic region on either side of the membrane. These proteins also have hydrophilic channels that allow substances to pass through them. The other type of proteins are peripheral proteins. Peripheral proteins are loosely attached to the membrane and often bind integral proteins. These proteins can make the plasma membrane stronger, transport materials through the membrane, participate in enzymatic activity, or help attach cells to other. The plasma membrane is a fluid mosaic model because the phospholipids are able to shift and slide past each other. They are able to do this because the bilayer is held together by weak hydrophobic bonds. This gives the membrane flexibility. Embedded proteins do not move nearly as much as lipids. If they do, they move very slowly. Steroids such as cholesterol affect the membrane in different ways based on the temperature. At a moderate temperature, cholesterol limits the fluidity by not allowing phospholipids to move much, but at low temperatures, it prevents the membrane from freezing by not allowing phospholipids to pack together tightly. Glycoproteins are carbohydrates that covalently bond to proteins, while glycolipids are carbohydrates that covalently bond to lipids. Both participate in cell-to-cell recognition. They allow cells to identify other cells by them. The membrane is selectively permeable due to only letting certain things through it. Only small molecules and ions can pass through the phospholipid layer freely. However, the structure of the bilayer prevents large molecules from passing through it. Proteins act like gates, only letting necessary particles through. All molecules and ions are able to move freely across the membrane. In addition, nonpolar molecules can easily dissolve across the membrane. Ions and polar molecules are not able to pass through the membrane due to the hydrophobic interior of the lipid bilayer. Water is able to cross the membrane through aquaporins. Aquaporins are channels that are specifically meant to let water pass through the membrane easily. The wall protects the cell and helps it to maintain its shape. The cell wall is much thicker, making it stronger than a cell membrane. The wall prevents the plant from taking in too much water, allowing it to function properly. Passive transportation is a naturally occurring phenomenon that does not require the cell to expend energy. The first type is osmosis. Osmosis is a movement of water through a semi-permeable membrane. The water moves from an area of high concentration to low concentration. Diffusion is very similar to osmosis. Diffusion is the movement of any substance into available space. Like osmosis, diffusion moves down the concentration gradient. Facilitated diffusion uses carrier and channel proteins to move particles across the membrane. These proteins that assist in diffusion span across the membrane. Active transport pumps solute against its gradient using ATP for energy. The sodium-potassium pump moves ions against their gradient. The sodium ion concentration is high on the outside of the cell and low on the inside, while the potassium ion concentration is low outside and high on the inside. The pump changes between shapes, pushing 3 sodium ions out and 2 potassium ions in. The different shapes are attracted to different ions. ATP provides the energy for the shape changes of the transport proteins. To move large particles across the membrane, endocytosis and exocytosis is used. Endocytosis engulfs the particles and packages it into vacuoles for use. Exocytosis releases a particle by a vesicle filled with the particle fusing with the membrane and releasing the contents.